So the last topic we'll talk about, we mentioned the word phylogeny a minute ago, and we said that it's the history of the evolution of a species or a group, especially in reference to the lines of descent and in relationships among organisms. The ways to establish phylogenetic relationships between organisms, first of all, include comparing the DNA or RNA sequences between the groups, comparing the amino acid sequences of specific proteins, or looking at the morphology, the body structure, shared derived traits. So oftentimes phylogenetic relationships are depicted in what are called phylogenetic trees or cladograms. So a phylogenetic tree is a branching tree-like diagram. Each node or point of divergence has two branching lines that come out, indicating an evolutionary divergence from a common ancestor. So a node is a point on that tree where a split happens. So that's a node. So we're saying that there's a common ancestor here, and that some of that common ancestor's descendants became, maybe we'll call this species A. Some of its ancestors became species B. In a phylogenetic tree, the length of the branches between each other represents time. Longer branches mean more time. On the other hand, a cladogram is very similar. But in a cladogram, length doesn't matter. Length doesn't indicate time. Other than that, phylogenetic trees are essentially the same. In AP Bio, we're going to use those terms to essentially mean the same thing. Phylogenetic tree and a cladogram. Think of it like an evolutionary family tree. So there's several important terms that you need to know related to cladograms. First of all, a clade is a group of biological taxa, maybe like a species, that includes all the descendants from a common ancestor. The root is the initial ancestor common to all the organisms in the cladogram. This is going to be the point which begins the cladogram. Morphology, we said a minute ago, is a branch of biology that deals with like the formation, the body structure of an organism. The ancestral trait is a trait shared by a group of organisms as a result of descent from some kind of common ancestor. A derived trait is a trait present in an organism but absent in the last common ancestor of the group. An outgroup is a group of organisms that serves as sort of a reference when we're drawing a cladogram. Essentially, the outgroup is hypothesized to be less closely related to the in-group than the in-group is to itself. And we'll see an outgroup in our phylogenetic tree in a minute. A node is a point on a cladogram where a split occurs and different species branch off from a common ancestor. It's important to note that in cladograms, the branches of the cladogram can be rotated around the node without changing the evolutionary relationships depicted in that cladogram. And then finally, a clade or a monophyletic group is a common ancestor and all of its descendants, all of the branches and nodes that branch away from that common ancestor. So here we can see a phylogenetic tree. So back here would be the root. This is the outgroup. This is the one that's most different from all of the other species. So this would be a species, species A, species B, species C, species D. This cladogram is showing us that they all evolved from some common ancestor back here. Here's a node. At this node, there's a common ancestor. Some of its ancestors evolved into species B. Some evolved into C and D. Here's a common ancestor shared between C and D. Some of its ancestors became species C. Some evolved into species D. Again, we can sort of see it drawn a different way down here, but the same relationships. So we've got a root. Here's a node. There's a common ancestor there. Some of that ancestor's progeny became species A. Some became species B. Right here's a node. Some of this common ancestor's progeny became C. Some went on to become either A or B. And again, here we can see the root, the beginning of the cladogram, the original common ancestor of everything else. Nodes are points where speciation happens. And again, some of the branches, we can hear them referred to as branches. We can see them drawn in this pattern or this more tree-like pattern. But wherever we go, there are nodes. And remember, nodes indicate the presence of a common ancestor. Now, the first cladograms were drawn based on morphology, based on the presence of shared physical characteristics. And you're going to do an activity later where you use morphology and a table called a character table to construct a cladogram based on morphology. But that's more the old way to draw a cladogram. Today, remember that our taxonomic system is based on evolutionary relationships and DNA. So today, most cladograms are built based on DNA comparisons. So they find certain genes or certain areas of introns and they compare the sequences. The reason that's a good idea is that essentially all organisms use DNA and RNA as their genetic material. It's universal. This means that the nitrogenous base sequences, that the sequences of A's and T's and G's and C's can be compared. Over the courses of millions of years, mutations accumulate. So that means the further back two species separated from each other, there's been more time for mutations in those genes to accumulate. So the more differences, that would mean the less related two species are. The less differences, that would mean the more related they are. Oftentimes when scientists look at molecular sequences, they use the non-coding gene sequences or the intron sequences. And the reason they choose those is because mutations in the genes oftentimes lead to changes that can cause death. And so the mutations aren't passed on as often in the coding, the exons. But the changes in the intron sequences aren't usually as severe. Organisms can live. Mutation rates are higher there. And we said a second ago, in addition to looking at gene sequences, like nucleotide sequences, you can also look at amino acid sequences. These change more slowly, though, than the gene sequences do, because remember that there are several codons that code for each amino acid. The genetic code is degenerate. So that means that there can be mutations without actual changes in the protein. So the best way to create a cladogram is to compare DNA sequences. More differences between the nucleotide sequences mean less related. Less differences mean more related. And again, you're going to do an activity in class where you build a cladogram based on both morphology and on molecular evidence. All right, that's it for our evolution discussion. Hope